In this video, we're going to use digital models and animations to explain how the embryo folds and how the internal body cavity forms as a result of that folding. So this was the digital model that we 3D printed and used in lab. I just want to remind you that at the margins of the developing ectoderm here, the amniotic membrane continues up to form a balloon above it, and that space inside that balloon is our amniotic cavity. And likewise, below the embryonic disc, we would have the umbilical vesicle, otherwise known as the yolk sac, which has been cut off here. So if we remove those, we're left with an embryonic disc that has an upper layer of ectoderm. This is technically the epiblast becoming ectoderm. If we remove that, we can see underneath that we've got a region of mesoderm that's accumulating, and then below that is the endoderm. So we're going to use some other models to look at the folding of the disc, but these are the three layers. This is the embryonic disc that we're going to use as a model to explain those processes. So this is the digital model that I built to animate the folding of the embryonic disc, and we're looking down onto the ectoderm. This is the location of the oropharyngeal membrane, which will eventually become the oral opening, and this is the location of the cloacal membrane, which will eventually form the anal opening. And if I rotate the embryonic disc, we can see that below it is a layer of mesoderm, and below that is the layer of endoderm. Now in lab, we mentioned that the embryonic disc folds like a burrito folds. And so what I mean by that is the two sides here are going to be brought together in the middle, and the two ends, the cranial end and the caudal end, will be brought together in the middle. And that's how you would wrap a tortilla around some delicious filling. You grab the sides and fold them together, and then finish by folding the other two ends. Now we're going to show you that animation in just a moment, but if I were to just animate that with the disc as is, you wouldn't be able to see anything that was happening in the middle of our embryo burrito model, as it were. So I'm going to be using two videos showing two different planes of section to show that folding. So one plane of section is a sagittal cut down the middle of that embryonic disc. And you can see that this sagittal cut shows us the locations of the oropharyngeal and cloacal membranes. And remember, that's where the ectoderm and the endoderm are fused together, and there's no mesoderm between the layers there. Now the other plane of section that we're going to use, and we'll start off with, is actually the coronal plane of section. This will allow us to see the lateral ends of the disc come together. And when we show the sagittal view, we'll be able to see the cranial and caudal ends of the embryo come together. And those two views will let us see all the interesting things that we need to understand about what's happening internally inside the embryonic structure as it folds. So starting with the coronal plane of section through this embryonic disc, we're going to be able to watch as the embryo folds. The lateral edges here and here will swing down to meet in the center. But we'll see where I put this plane of section, we're not actually going to see them completely meet, because remember that this endoderm layer is connected to the yolk sac. So our plane of section is right through the location where that's happening. So let's go ahead and play this. There they go. Sides are folding down. And the endoderm is curling in towards itself, starting to create a tunnel within this embryo. That is not the body cavity. Within that endoderm layer, we're actually forming the gut tube. And there we go. So we've seen the edges come down around, And what we want to take away from this is that the ectoderm is now forming the outer covering of the embryo all the way around it. And the endoderm is coming together to form a single tube that's running the length of the embryo. And this little continuation down here will eventually get closed off as the umbilical cord develops. So the communication between this gut tube and the yolk sac below it will eventually get closed off, leaving us just with our digestive system here. So let's now take a look at the same process happening, except we'll do the sagittal cut and watch the ends come together. So I've now loaded the other animation, which is the sagittal plane of section through this embryonic disc. From this sagittal view, we can once again see ectoderm is forming this top layer, endoderm is forming this bottom layer, and then there's a region of mesoderm stretching between those two. We also can see the oropharyngeal membrane and the cloacal membrane, which are these locations where the ectoderm is fused to the endoderm, and there's no mesoderm in between. Now that's important because we can now see that the mesoderm is in these three different locations. And what's important for this week's lab is that the heart is going to develop from this region of mesoderm. And remember that the neural tube was brought into the embryonic disc, and it's not modeled in my demonstration, but that neural tube is actually running in this zone of mesoderm from here to here. It was limited between these 2 points. And if we follow the folding of this embryonic disc, one of the important consequences is that region that's forming the heart is getting pulled down and around. And the cranial end of the neural tube, which is located here, is rotating around. So that brain that develops and that heart that develops are kind of swapping places in the order of the embryonic disc. There we go. So this is that cardiogenic area, and this is where the cranial end of the neural tube is. So this is where our brain's forming. And so now we have the proper sequence of structures. Head with brain, opening of the oral cavity, and heart. Now we can also see that the digestive system is forming here, stretching between the oral opening and the anal opening. And this location here is where the endoderm is attaching to the umbilical vesicle, which I also don't have in this model. But eventually this will close off, creating a continuous digestive system from end to end. Okay? This is also the location of where the umbilical cord is going to be forming. So this is going to stretch to the placenta and keep this embryo alive by that fetal circulation. So the end result after both lateral and cranial caudal folding is that you turn that flat embryonic disc into this structure. This is our three-dimensional embryo with the head developing here. The cranial end of the neural tube is forming the brain in here. We've got the oral opening forming here at the oropharyngeal membrane. This bulge of tissue is that part of the mesoderm where the heart is going to develop. We have the cloacal membrane back here, which will eventually develop into the anal opening. And we have the communication from our gut tube to the umbilical vesicle, or otherwise known as yolk sac, which will eventually get closed off, and the umbilical cord is going to be developing in this mesoderm right here. We also have the amniotic sac attached still at the ectoderm. It's not in the model, but that will completely surround the embryo so that this structure is actually suspended within inside an amniotic cavity. Now, what we didn't explain with the animations of the embryo folding is how the internal body cavity is formed. So to explain that, I'm going to go back to my embryonic disc and I'm going to show you something that's happening in the mesoderm. So we've just seen how the embryonic disc folds, but we haven't seen how the internal body cavity is formed. To understand that, let's go back to this model of the flat embryonic disc before any folding occurs, and let's go back to that coronal section. Now we've been talking about the ectoderm, the endoderm, and the mesoderm, but this is a simplified model. We have to remember that this is after neurulation. So the neural tube is actually now developing in this zone at this location. And below it, the notochord would be situated here. So there are many other things that are developing within this embryonic disc even before it folds. And one thing that is changing at this time is that a space is forming here in what we call the lateral plate mesoderm. And there's a space on the other side as well. And these two spaces are really actually just one continuous space. It's being sectioned in two different places. And I can put in a model of that single space like so. And if I return to that top-down view, we see that it's horseshoe-shaped. We call this space the intraembryonic coelom. When I put the rest of the embryonic disc back, we can understand that that coelom is passing in front of the oropharyngeal membrane. And if I switch to my sagittal section, we can see that the very front of the intraembryonic coelom happens to be traveling in front of the area which is going to be producing the heart. Remember, this part of the mesoderm forms the heart. Now I can actually remove this model from my embryonic disc and create a space here which represents that intraembryonic coelom. There we go. And by creating the actual space in my model, we can now see that it's forming a tunnel which would be extending through the embryonic disc like so and exiting out the side. If I turn around, we can see where it's emptying out. And that's symmetrical on both sides of the embryonic disc. Now, if I return to my coronal section, we can now see that the lateral plate mesoderm is actually being divided into two halves. This upper half, which is attached to the ectoderm, is our somatic mesoderm, which is going to be innervated by somatic afferent nerve fibers. And we call the ectoderm and somatic mesoderm together the somatopleur. The other half of lateral plate mesoderm down below the intraembryonic coelom is the visceral mesoderm, and it's going to have visceral afferent nerve fibers growing into it and innervating it. And it's attached with the endoderm here. Together, this double layer is called the splanchnopleur. Now, when the disc folds, that space inside it will follow the folding. And over the next few minutes, I'm going to show how the folding of the embryo changes the shape of that space inside to produce the internal body cavity. So what we're looking at here on the left is that same model of the intraembryonic coelom, that space that is running through the mesoderm of the developing embryonic disc. And this is before any folding occurs. And on the right, we see the shape that it forms after the embryo folds. Now, right away, we can see two things. First, after folding, the spaces become more complicated. And that's because the embryo itself is more complicated in shape compared to the flat embryonic disc that produced it. Second, we can also see that this space has comparatively gotten larger. It's gotten longer and it's gotten thicker. And that's because as the embryo develops, more and more cells divide and it overall gets larger, and the structures inside that embryo get larger. And that means that the spaces between those structures also gets larger. Now, I'm going to rotate the view so that we can look a little bit more closely at some of the changes that have occurred. Okay, this is once again the cranial end of this intraembryonic coelom, and this is further back in the embryo. And if we look at the cranial-most end, this location here is going to be subjected to the cranial folding of the embryo in this direction, as well as the lateral folding in this direction. So this region here gets squashed on itself. So it gets rotated back and folded over and folded in from the sides. And so you end up forming this region here, which is going to be our developing thoracic cavity. Right? So we get this nice large volume that's going to become our thoracic cavity, including the pericardial space. Now, second, the lateral folding of the embryo. So as this side folds down and this side folds down and they meet, when you're further back in the embryo, that's going to bring these sections together, which are going to end up fusing together. And so you end up creating a single space across the midline of that developing embryo. And that is corresponding to our abdominopelvic cavity. And it continues forward all the way to here. This interruption is once again where that yolk sac is connecting to our digestive system. That will also close off, producing a single abdominopelvic cavity. Finally, if we look between the developing abdominopelvic cavity and the thoracic cavity, we have two extensions corresponding to these two columns of our early intraembryonic coelom. And these tunnels are connecting the thoracic cavity with the abdominopelvic cavity, and they're called the pericardio-peritoneal canals, because our pericardial space forms in here and our peritoneal space forms in here, and that's where the name comes from. If you haven't heard those terms before, we'll use them again when we do abdominal anatomy, but they're basically just subdivisions of those two spaces. So we've got now a more complicated shape, but it's really just produced by folding this horseshoe and smushing together certain parts of it. Now let's integrate that folded intraembryonic coelom shape in with our model of the embryo. So to reorient, this is that sagittal section through the embryo after the disc has folded. All right, here's that nice view from the side where you can see the bulge of the head, and down here is that caudal end. And returning to our sagittal cut in the developing head region, this is where the brain will develop. You'll have the spinal cord extending down. This region of mesoderm here is where our heart will be developing. And our gut tube now connects the oropharyngeal membrane with the cloacal membrane. And it will undergo further differentiation and development to produce our pharynx and stomach at some point, and large and small intestines at another point. So that's the body plan now. And if we add in our intraembryonic coelom, after it's been folded, we can see that our thoracic cavity region here is lining up with that part of the mesoderm where the heart develops. And our abdominopelvic cavity is surrounding, remember there's another side to this, is surrounding the developing gut tube. And connecting them are those pericardioperitoneal canals. And what I can do is I can remove the mesoderm where this space is, just like I did on the earlier model. There we go. We've now introduced the actual intraembryonic coelom into this digital model. So this is our developing thoracic cavity right next to where the mesoderm is producing the heart. And here we have abdominopelvic cavity space, which is surrounding that developing gut tube. And if I rotate it and we look up through the thoracic cavity, we see that there is a pericardioperitoneal canal that is connecting the thoracic cavity space here with the abdominopelvic cavity space here. So there we go. We have our internal body cavity. Now we know that these spaces aren't connected in the adult, and they'll be closed off when mesoderm from this region grows up in a structure called the septum transversum to block off that canal. And the diaphragm is going to develop in large part from that septum transversum. And that's the barrier that we have in the adult human between these body cavity spaces. Okay, now one last thing. Let's take a look at a coronal section through this embryo after it's folded and after the intraembryonic coelom has been merged together at the abdominal level. Okay, ignore these geometry artifacts, some problem with the software. But what I want to emphasize in this view is remember that the lateral plate mesoderm is what's folded around on either side here. And the intraembryonic mesoderm was separating it into visceral mesoderm and somatic mesoderm. And so we can see how both sides of the visceral mesoderm have joined to follow around that completed gut tube, and both sides of somatic mesoderm have now joined in the middle. And it's these two regions of mesoderm separated by this space that then get different types of sensory fibers growing into them in large part. So you get the somatic afferents preferentially in here, and the visceral afferents innervating the visceral organs.